Hello everyone, welcome back to the session. So in today's class we are going to discuss about a cellular respiration. What are all the most important steps which are involving in cellular respiration, because cellular respiration is an umbrella term. So in this term we are having different types of steps, and what are all the different cycles which are involving in this cellular respiration, everything we are going to discuss in detail. Okay. Now, see here. So the cellular respiration, just look at the picture very carefully. So this is the cellular respiration, and what are all the organelles we can see which are involving in this particular cellular respiration. The first important thing is a cytosol and the mitochondria. In the mitochondrial matrix, there we can see the citric acid cycle, and in the cytoplasm we are seeing a glycolysis. So the glucose is the one converting into a pyruvate, and this particular step is called as the glycolysis process, and the glycolysis is taking place in the cytosol. So see, this is the first most important thing you have to keep in mind. So cellular respiration, it is not at all a single step, it is a multiple step, multiple cycle. For all those multiple cycle, different organelles are going to involve in this. We are going to discuss everything. Just look at this picture very carefully. So this is what a cytosol, and in the cytosol the glucose is converting into a pyruvate with, and this cycle is called as a glycolysis. So you should know the glycolysis will be taking place in the cytoplasm. And so in the mitochondria we are having a outer membrane, inner membrane, and the cristae and matrix. So these are all the structure of the mitochondria. So in the mitochondrial matrix, and another important cycle is going to take place, and that cycle is called as a citric acid cycle. So in the citric acid cycle, so this is, see here, and this is one of the cycle, and glycolysis is another cycle. And again the glycolysis, this is also said to be a glycose if the glycolysis, if the glucose is converting into a pyruvate. So what is the difference between these two glycolysis? Because is in the sense, so in the glycolysis will be taking place in the aerobic respiration, and the glycolysis which is taking place in the anaerobic respiration. So this is the aerobic respiration, this is anaerobic respiration. So with the presence of oxygen, this glucose is converting into a pyruvate, and in the absence of this oxygen, this glucose is converting into a pyruvate, and this pyruvate is converting into a lactic acid and ethanol in the absence of oxygen, and this process is called as a fermentation. Now this lactic acid or ethanol is again in the anaerobic respiration. This is in the aerobic respiration. So this is what a basic understanding, and this is not the end point of the cellular respiration. Some more important things is also going to take place. This is the simpler picture of the cellular respiration. Now just like I said, in the aerobic respiration of glycolysis and the anaerobic respiration, just look at this picture. See, the glucose is converting into a pyruvate, and the pyruvate is entering into a mitochondria. There they are producing 36 molecules of ATP. So just keep in mind, so in the aerobic respiration, how many number of ATP is produced? 36 ATP is produced. So how this 36 ATP molecules is produced, it will be clearly discussed in the cycle. Just as of now, know that this aerobic respiration, in this aerobic respiration, the glucose is converting into a pyruvate, and this pyruvate enter into a mitochondria, and undergoing some cycles, it is producing a 36 ATP molecules. And what is with respect to this anaerobic respiration? Will explain you. Now see, so this is what a cellular exact picture of a cellular respiration. See, this is the first cycle, that is glycolysis cycle. So in the glycolysis, the glucose is converting into a the glucose is converting into a pyruvic acid. Now this pyruvic acid is entering into a mitochondria, is undergoing a Krebs cycle, and in the Krebs cycle again they are producing an ATP, energy molecule, and again they are the one entering into the electron transport chain. So the electron transport chain, oxidative phosphorylation, and this glycolysis Krebs cycle, all these together we are saying that as a cellular respiration. So let me explain you little bit detailed manner. So first of all, what is mean by cellular respiration? You know, everything in our body is a metabolism. Like in the same way, the cellular respiration, that is the respiration, is also a metabolism. It is a metabolic process by which the cells break down organic molecules to release energy and convert into an ATP, the universal energy currency of a cell. See, this is a metabolic process by which the cells break down organic molecules to release energy. See here, and some process which are involving in breaking the complex molecules into simpler molecules. Whenever it is breaking, they are releasing an energy, and that particular energy is converting into an ATP, and this ATP is called as an adenosine triphosphate, and this phosphate is nothing but an energy molecule, and this is why this ATP is considered as a universal energy currency of a cell. Now, to put in a nutshell, so any complex molecule will be breaking into a simpler molecule. As a result of this breakage, we getting some energy, and this energy is getting converting into an ATP, and this will be the universal currency of a cell. So, in the means of respiration, cellular respiration, they are producing an energy. So, this is what a cellular respiration. Now, so this cellular respiration is one of the most fundamental biological process in living organisms. See here. So what do we have discussed? So in the cellular respiration, they are involving in breaking the complex molecules. As a result of breaking, energy will be released, and this energy is converting into an ATP. And why we are in need of energy? So this is why we are saying the cellular respiration is a fundamental process, because they are the ones supplying energy for movement, for active transport, biosynthesis, cell division, maintenance of a homeostasis. So as a result, we can say that for energy is required for all of the basic functions to the most important functions. Energy is a basic unit. So for the production of this energy, cellular respiration plays a crucial role. That's why this is a fundamental biochemical process. See, and how can we define the cellular respiration? See here, cellular respiration, it is a series of enzyme control oxidation reduction reactions in which glucose is broken down to produce ATP with carbon dioxide, oxygen as end products in aerobic respiration. See, what do we have discussed? A complex molecules like glucose is converting into a simpler molecules, and they are the one providing us an energy. Now, this energy, how this glucose will providing an energy with the help of certain enzymes. It is not one or two enzymes, many enzymes. That is why we are saying series of enzyme controlled reaction, and that reaction, either it may be an oxidation reaction or it may be a reduction reaction. Okay, so either the both of the reaction will be taking place in this enzyme controlled reaction. So as a result, what are the byproducts as a result of this conversion? This carbon dioxide and water is as a end product. So this will be acting as a byproduct, released as a byproduct in case of aerobic respiration. So this is how we can define the cellular respiration. Now what will be the overall reaction of this cellular respiration? So see here, C6H12O6, this is a glucose. Glucose is combined with the oxygen. How many molecules of oxygen? 6 molecules of oxygen. See, glucose, one molecule of glucose with 6 molecules of oxygen, together they are forming 6 carbon dioxide, 6 water molecule. Both are the end product or the byproduct, and the release of energy, and the energy in the form of ATP. So glucose here will be a fuel molecule. Oxygen, it will be a final electron acceptor. So this can be expected in our exam. Who will be the acceptor of an electron? Why the electron will be released, and why the released electron has to be accepted? Everything will be discussed detailedly in the electron transport chain, and that will be the one of the most important step under the cellular respiration. Now, as a result of this, ATP is going to produce. So, you should know this will be the overall reaction of this aerobic respiration, not anaerobic respiration. Now, so we are having two different types of cellular respiration. One is aerobic respiration and another one is anaerobic respiration. So, in the aerobic respiration, we are in need of the oxygen. In aerobic respiration, we are in need of the oxygen, and here a molecule is getting oxidized. A molecule is oxidized, and as a result of this oxidation, we are having a carbon dioxide and water and ATP. See here, you should know only whenever, see how the digestion and respiration will be taking place in a cooperative manner. See, we are taking an oxygen from the atmosphere is completely related to the respiration process, and the carbohydrates has to be broken down into the simpler molecules that will be completely related to the digestion. So you should know that whenever some molecule has to be digested, it definitely needs an oxygen. So with the help of this oxygen, complex molecules like glucose will be breaking down and to give an energy. Why we are intaking a food? To get an energy. How the energy will be released? With the help of the cellular respiration. So this is what the simplest explanation as I know. See here, aerobic respiration, definitely it needs an oxygen, and just remember, oxygen is required to a complete oxidation of a glucose. As a result of this oxidation of glucose, we get an energy, and not only energy, we are also getting some end products like carbon dioxide and water. So how many molecules of ATP is going to release per glucose in eukaryotes? In eukaryotes, per glucose we are getting approximately 32 to 32 ATP. So this is very important. Now anaerobic respiration, and this anaerobic respiration is also called as a fermentation. And the first and foremost important point with respect to this anaerobic respiration is it doesn't need any oxygen. Without oxygen, do energy production is possible? Of course it is possible. We are going to discuss about that. See, without oxygen, of course the glucose can be broken down, but partial breakdown of the glucose. So in case of an aerobic respiration, there is an involvement of oxygen. As a result, there will be a complete oxidation of glucose. Now in anaerobic respiration, there will be a partial breakdown of glucose. Now what will be the end products? Lactate. Lactate in case of animals, and ethanol plus carbon dioxide in case of yeast, which means glucose is converting into a lactate or ethanol. Lactate in case of animals, ethanol in case of yeast. So this will be the end product of the anaerobic respiration. So you should know, anaerobic respiration, there will be no involvement of oxygen, and there will be a partial breakdown of glucose, and it is also called as a fermentation. And these are all the end products: lactate and ethanol, carbon dioxide, all these were the end products based on the different animals. And the most important difference between aerobic and anaerobic respiration is how many molecules of ATP per glucose will be produced. See, in case of aerobic respiration, we have studied approximately 30 to 32 molecules of ATP will be produced per glucose in case of aerobic respiration. Just look at this. In anaerobic respiration, how many ATP will be produced per glucose? Only 2 ATP. So this is very important. So when compared to aerobic respiration, anaerobic will produce very less number of ATP per glucose. So this can be expected in our exam. See here, just now we are entering into the most important part. So how many stages are involving in the cellular respiration? So only when all these four major stages have been completed, we are saying that as a cellular respiration. Okay, so now we have studied that four major stages have been present in the cellular respiration. What are all those stages? First important stage will be the glycolysis, and the second stage will be the pyruvate oxidation, and the third step will be the Krebs cycle. It is also called as a TCA cycle. TCA cycle in the sense tricarboxylic acidic cycle. tricarboxylic acid cycle, and electron transport chain, that is ETC. All together we are calling them as a cellular respiration. See, whenever we are studying about the stages of the cellular respiration, you also know that what each stage will contribute for, which means what is their function of each and every stage and where all these stages is going to take place. So the location and the function of each stages is very important. We can expect in our exam. See, glycolysis will be taking place in the cytoplasm, and the main function of this glycolysis is glucose is converting into a pyruvate. Glucose is converting into a pyruvate, and pyruvate oxidation. See, this is a pyruvate oxidation and the location where the pyruvate oxidation will be taking place. So we can say this as a pyruvate oxidation or pyruvate decarboxylation. Both are the same reaction. Same reaction but in a different meaning. Different name but the same reaction. See, the pyruvate oxidation will be taking place in the mitochondrial matrix region. It is in the mitochondria, exclusively in the matrix region, and here the pyruvate will be converting into an acetyl-CoA molecule, and in the Krebs cycle, which is also called as a tricarboxylic acid cycle, again it is also taking place in the mitochondrial matrix region. Here the complete oxidation to carbon dioxide, how it is taking place, everything will be discussed. And in the electron transport chain, this can be expected in our exam. This ETC will be taking place in the inner mitochondrial membrane. So the inner mitochondrial membrane, here finally the ATP will be produced. The ATP will be produced via which process? That is via the oxidative phosphorylation. Via the oxidative phosphorylation, ATP will be produced. That is, we are saying that as an electron transport chain that is taking place in the inner mitochondrial membrane. This table is very important. Kindly make a note of it. Now, so what? Just let me give you a basic idea or overview of all those four stages. So in the glycolysis, the first important thing will be, it will be occur in the cytosol. It will be occur in the cytosolic region. And what exactly in the glycolysis will be taking place? The glucose molecule will be taking place into 2 pyruvate. Glucose, 1 glucose will be converting into 2 pyruvate. What will be the net gain? That is 2 ATP and 2 NADH. 2 ATP and 2 NADH, this will be the net gain. So this can be expected in our exam. How many ATP will be produced here? See, you should know the difference. Here 4 ATP will be produced and 2 ATP will be used in the cycle itself. As a result, only 2 ATP will be the net gain. So this exact information, exact cycle will be discussed whenever I explain you the glycolysis detailedly. Just as of now, know what are all the most important thing taking place in the glycolysis. So the next important reaction will be the pyruvate oxidation, or we can say pyruvate decarboxylation. Just not most precisely, we can say this as a pyruvate decarboxylation. Why we are saying decarboxylation? Because of the removal of carbon dioxide, we are saying decarboxylation. And you should know the link reaction between the glycolysis and the Krebs cycle will be this pyruvate decarboxylation. So this pyruvate decarboxylation will be called as the link reaction. And where this exactly taking place? This occurs in the mitochondrial matrix. So the mitochondrial matrix region, we can see this pyruvate decarboxylation. Now, see what do we have discussed? In the glucose, in the glycolysis, glucose will be converting into 2 pyruvate. Okay, so just know that the glycolysis will be converting into a pyruvate in case of glycolysis. Glucose will be converting into a pyruvate, and this pyruvate will be converting into acetyl-CoA. Whenever the pyruvate is converting into an acetyl-CoA molecule, and they are releasing the carbon dioxide. They are releasing the carbon dioxide and they are producing the NADPH. And per glucose, for 1 glucose, 2 pyruvate will be produced, and this reaction is called as a glycolysis. And 2 pyruvate will be converting into 2-acetyl-CoA, and they are giving out 2 carbon dioxide and they are producing 2 NADH. This will be the product of 1 glucose. This also can be expected in our exam. Now the third important cycle will be the Krebs cycle. It is also called as a citric acid cycle, also called as tricarboxylic acid cycle. That is nothing but TCA cycle. Now, as we have studied before, this is taking place in the mitochondrial matrix, and the acetyl-CoA is completely oxidized. Acetyl-CoA here, it will be completely oxidized. So, you should know for 1 glucose, it is producing 4 carbon dioxide, 6 NADH and 2 FADH and 2 GTP. Here they will not produce ATP, but instead of ATP they are producing a GTP. Again, this is also a energy molecule. So this also can be expected. How many carbon dioxide will be produced? 4 carbon dioxide and 6 NADH, 2 FADH, 2 GTP. So this can be expected in our exam. Kindly make a note of the end product. Now the fourth important stage will be the electron transport chain. So what do we have discussed? This electron transport chain will be occur in the inner mitochondrial membrane, and here who is the one donating an electron? Here NADH and FADH2, which is produced in the Krebs cycle, they are the one donating an electron, and as whenever the electron will be released, as a result of the release of these electrons, they are producing a proton gradient. As a result of this proton gradient, so whenever the electron is released, there will be the release of energy, and that energy, with the help of this ATPase enzyme, they are producing an ATP. Approximately how many ATP will be produced? 26 to 28 ATP molecules will be produced in the electron transport chain. Now, see, this is very important in the cellular respiration. See here, glycolysis. So in the glycolysis, 2 ATP will be produced. This will be the net gain. And here, Krebs cycle. In the Krebs cycle, we are having 2 ATP molecule will be produced in the Krebs cycle. That is 2 GTP will be produced. NADH and FADH via electron transport chain, they are producing 26 to 28 ATP, and finally total 30 to 32 ATP will be produced per glucose. This is very important in the ATP production in each of the cell respiration stages. Now, what are all the most important concepts which are involved, which are seen in the cellular respiration? First important thing will be the oxidation and reduction reactions. Oxidation and reduction reactions. Now here glucose is completely oxidized, oxygen is reduced to water. So this is major important thing. See here the oxygen, see, first you should know what is meant by oxidation and what is meant by reduction. Let me explain you very clearly. So this is oxidation and this is reduction. Okay, so in the oxidation reaction, any molecule, if it accept the oxygen, this is called as an oxidation. If removes the hydrogen, if removes the oxygen, then it is called as a reduction. It is accepting an oxygen, it is releasing the oxygen. So that is what an oxidation reaction, reduction reaction. And the second important point will be, if they accept the hydrogen, that is called as a reduction reaction. And if it releasing the hydrogen, it is called as an oxidation. And last but not the least and the most important point, when they are accepting an electron, they are called as a reduction reaction. When they are releasing an electron, they are called as an oxidation reaction. Most important thing. See, oxygen acceptor, oxidation, oxygen donor, reduction reaction. And if any reaction, if it accepts the hydrogen, reduction reaction, donating the hydrogen, oxidation, not accepting the hydrogen, that is oxidation. If it involving in gain of the electrons, then it is a reduction reaction, and if it losing the electrons, it is called as an oxidation reaction. So these are all the oxidation and reduction reactions difference. Now, the second important role will be the role of electron carrier. So, what are all the important function of this electron carrier? So, there will be two major enzymes which are involving in which are acting as an electron carrier. See, NAD plus. NAD plus, they are whenever you find any plus or minus in any molecule, that is in an oxidized state. So this NAD plus is acquiring a hydrogen. So just I have explained you in the previous slide, if any compound or if any molecule, if it is accepting a hydrogen, then we are calling them as a reduction reaction. So NAD plus, they gives rise to NADH. So this is very important. Now FAD, again this FAD is also accepting a hydrogen, then it is also undergoing a reduction reaction. So see here, they are gaining an electron, they are also gaining an electron. So this is why we are saying that as an electron carrier. So the most important coenzymes which are involving in accepting the electron, which are carrying the electron, is NAD plus and FAD. They carry high energy electrons, deliver them to electron transport chain. See here, they carry high energy electrons. They are not carry one or two electrons. They are high energy electron carrier, and they are the one, they are donating all these electrons to the electron transport chain. So we'll discuss what are all the electron transport chain, which means. Now, what are all the different types of ATP will be produced? So, we are having two different types of phosphorylation. One is substrate level phosphorylation and oxidative phosphorylation. In both these levels of phosphorylation, phosphate group is going to produce. We are going to get an ATP. We are get an ATP. But in which means that in which mode, in which medium the ATP will be produced. So based on the source where we can get the ATP, we can categorize the ATP production into two, that is substrate and oxidative phosphorylation. In both of these phosphorylation, we are going to get this ATP. Now what will be the difference? Direct transport of phosphate to ATP. Directly the phosphate group will be converting into an ADP. It will be occurs in the glycolysis and Krebs cycle. See, in the cellular respiration, we are having four important stages: glycolysis and Krebs cycle and electron transport chain and pyruvate decarboxylation. So all these four stages together we are calling them as a cellular respiration. And in the substrate level phosphorylation will be taking place in the glycolysis and Krebs cycle. Here the phosphate group directly transferred to the AMP. See here, adenosine monophosphate. This AMP, while they are accepting one phosphate group, they are converting into adenine diphosphate. So direct transfer of this phosphate group. Now oxidative phosphorylation, here the ATP will be produced via electron transport chain. The ATP will be produced via the electron transport chain, uses the proton gradient. By using the gradient of this proton, they are producing an ATP via the electron transport chain. We are going to see what are all those phosphorylation. Just as of now, know what will be these words. Now, see, so in aerobic respiration, oxygen is required. In anaerobic respiration, the oxygen is not required. And the breakdown, here the breakdown will be complete breakdown of the glucose. Here the partial breakdown of the glucose. Here the end product will be the carbon dioxide and water. And here it will be the lactate and ethanol. The ATP here it will be 32 ATP, and here it will be 2 ATP. Location here the cytosol and the mitochondria. In anaerobic respiration will be taking place only in the cytosol. So we'll discuss this respiratory quotient. Now see here, respiratory quotient means the ratio of number of carbon dioxide released and the number of oxygen consumed during the reaction. See, in a reaction, how many number of oxygen is consumed for to take up the to take up the oxygen, how many carbon dioxide released for number of oxygen. So the ratio between the carbon dioxide released and the oxygen consumed is called as a respiratory quotient. So by see the RQ value of these biomolecules, that is carbohydrates, fats and lipids, the respiratory quotient value will be higher for the carbohydrates, which means what? The carbon dioxide released will be 1. If for one oxygen molecule, for one carbon dioxide released, for one oxygen molecule will be consumed. So as a result, the RQ value will be 1. So the fats means what? See, the number of our carbon dioxide released will be lesser when compared to the oxygen consumed. So more oxygen is needed to break down the fats. So when compared to the proteins and fats, the RQ value will be higher for the protein. So we can say that as a, see, fats are carbon dioxide RQ value will be higher, which is in the normal, and in the fats they are having 0.7 and the proteins they are having a 0.8. So this can also be expected in the question, and we'll discuss this after we'll finish the glycolysis. Now we are going to see the glycolysis reaction. See, what are all the steps which are involving in the glycolysis? Now just look at this picture. So this is the glucose. This is the glucose. This glucose, with the help of this hexokinase, they are converting into glucose 6-phosphate. Okay, see here, glucose means how many carbon? 1, 2, 3, 4, 5, 6. We are having 6 carbon in the glucose. What is meant by glucose 6-phosphate? At the 6th carbon, the phosphate group has been added. See, whenever any enzyme has been ended with the word kinase, that enzyme is involving in adding the phosphate group. Okay, let me explain you once again. If any enzyme which ends with the word kinase, if any enzyme ends with the word kinase, that enzyme will definitely involving in adding the phosphate group. So that's why we are segregating that particular enzyme under the this particular group. See here, now as it is a hexokinase, hexo means what? 6 carbon. So glucose is also having a 6 carbon, and at the 6th carbon they are adding a phosphate group under which enzyme? Hexokinase enzyme. Now the second thing will be this glucose 6-phosphate will be converting into the fructose 6-phosphate. So the number of carbon is similar for both glucose and fructose, and at the 6th carbon 6-phosphate group has been added, so that's why fructose 6-phosphate. Now which enzyme is mediating this particular difference? See here, phosphoglucose isomerase. So isomer means same structural formula but different molecular formula. Okay, so see here, same structure, both are in the same structure but different molecular formula. So C6H12O6, both for the glucose as well as the fructose, but their molecular structure will be something different because of the presence of a functional group. Now fructose 1,6-bisphosphate, here not only at the 6th carbon, in the first carbon they are adding a phosphate group, so that's why 1,6-bisphosphate. This is nothing but bi means 2. At 2 sides, that is at the first carbon and the 6th carbon, they are adding a phosphate group. That's why the name fructose 1,6-bisphosphate. Now with the help of which enzyme? Again any enzyme will end with the word kinase. Here phosphofructokinase. So this is very important. Now at this stage, this reaction will be divided into two branch. That is dihydroxyacetone phosphate DHAP and glyceraldehyde 3-phosphate G3P. Now this see here, this is the area you have to keep a more attention. See, this glucose 3-phosphate will be converting into 1,3-bisphosphoglycerate, and 1,3-bisphosphoglycerate is converting into 3-phosphoglycerate, 3-phosphoglycerate, 2-phospho, and phosphoena and pyruvate. So this is how the glyceraldehyde 3-phosphate will continue the cycle. Just like in the same way, this dihydroxyacetone phosphate will also converting into 1,3-bisphosphoglycerate. Now 1,3-bisphosphoglycerate will be converting into 3-phosphoglycerate. Now 3-phosphate to 2-phosphoglycerate and 2-phosphoenol pyruvate and pyruvate reaction. So what are all the thing you are discussing here, you are seeing here, this has been converted by both dihydroxyacetone phosphate separate and this glyceraldehyde 3-phosphate in a separate. Okay, so this will be common for both. You don't think both are combining and they are producing the sequence. No, it is not. Dihydroxyacetone phosphate is converting like this, and the glyceraldehyde 3-phosphate is also converting like this in a separate manner. Now, see here, glyceraldehyde 3-phosphate will be converting into 1,3-bisphosphoglycerate with the help of this G3P dehydrogenase, which means glyceraldehyde 3-phosphate dehydrogenase, which means what? At the first carbon also they are converting in, they are adding a phosphate group, so that's why 1,3-bisphosphoglycerate. Now phosphoglycerate kinase, again 3-phosphoglycerate will be converting from the 1,3-bisphosphoglycerate. Now phosphoglyceromutase, as a result they are converting into 2-phosphoglycerate. With the help of this enzyme enolase, they are converting into phosphoenol pyruvate. Finally pyruvate kinase will be converting into a pyruvate. See, so for this, in this cycle you should know, see, and in this cycle you should know what are all the compound which is converting into which compound and with the help of, with the influence of which enzyme. And in this, the same thing, I don't want to merge everything in one single cycle. That's why I have separated into 3 cycles. In each cycle everything is a glycolysis but with a different information. Just look at here, up to this, which means wherever it is converting into 2 branches, up to this, that is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, up to that reaction, that is an enzyme energy investment phase, that is preparatory phase, enzyme investment phase, which means the energy is investing there. Which means, see here, the ATP is converting into ADP, one phosphate molecule will be released and that phosphate molecule will be added in the first carbon. So that's what done by this fructose 1,6-bisphosphate. This is what happening there. See now, here these are the energy investment phase and the preparatory, which is also called as a preparatory phase. Now finally the pyruvate has been produced by both glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, and this phase is called as an energy payoff phase, which means what? We are investing and there is some point we are going to take the investment back. So that will be the payoff phase, that is payoff phase. Okay. Now see here, the net gain. See, just look at this. This is very important. This will be a questionable area. 4 ATP will be formed. Okay. So in the glycolysis, 4 ATP will be formed. So in the 4 ATP, just listen to me very carefully. 2 ATP has been used in the cycle itself. So 4 ATP minus 2 ATP. So that finally 2 ATP will be produced. So this will be the net gain. So this will be the net product. So if in the question, if they ask a gross value, then you should know 4 ATP. Gross means total ATP. Okay. Now in the net ATP means what? Those ATP which are used in the respiration process, that will be 2. So 4 minus 2, it will be 2. So 1 glucose will be converting into 2 pyruvate. And in the glycolysis itself, 2 ATP will be produced, 2 NADH will be produced. So you should know that 1 NADH is equal to 3 ATP. 3 ATP, and here it will be 2 NADH, then 2 into 3 is equal to 6 ATP. So together here it will be 8 ATP. Now the same thing. Now glycolysis, see here, glycolysis, how can we define? See here, it is a pathway, it is converting a glucose into pyruvate with the production of ATP and NADH. ATP and NADH. So in both prokaryotic cell and in the eukaryotic cell, this glycolysis will be taking place in the cytoplasm, and this will be the first step of the cellular respiration. Now, basic overview, this glycolysis in Greek term, this glycs means wheat, lysis means splitting, which means nothing but carbohydrate is getting splitting off. What will be the pathway? It is also called as, this glycolysis is also called as Embden-Meyerhof-Parnas pathway, that is Parnas pathway, it is EMP pathway. So it is located in the cytosol. And what will be the oxygen requirement? So this glycolysis will be taking place both in the aerobic as well as the anaerobic respiration. Now, so the glucose, the starting molecule will be 6 carbon containing compound, just now I explain you in the picture. And what will be the end product? 2 pyruvate, and each will be 3 carbon. So just look at this here, 6 carbon will be the glucose, and this will be converting into pyruvate, 2 pyruvate, each pyruvate they are having a 3 carbon, so 3 plus 3 is 6, the equation balanced. And whenever this 6 carbon is converting into 2 molecules of pyruvate, they are producing 2 ATP and 2 NADH. Very, very important. Now, 2 phases of glycose, and there is an energy investment phase, that is preparatory phase, and another phase will be the payoff phase. And in the energy investment phase, they are using 3 ATP. They are using 2 ATP. How they are using? See, glucose will be converting into 2 molecules of glyceraldehyde 3-phosphate. In this energy preparatory phase, which means what? Glucose will be converting into glucose 6-phosphate. Glucose 6-phosphate will be converting into fructose 6-phosphate. Fructose 6-phosphate will be converting into fructose 1,6-bisphosphate, and in this fructose 1,6 will be converting into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Both are the isomer. Both are the isomer in the sense what? Both are having the same structural formula but different molecular formula. That's why they are saying 2 molecules of glyceraldehyde 3-phosphate. So up to this stage, we are saying that as a preparatory phase. In this preparatory phase, 2 ATP molecules will be used. Now, see here, phases of glycolysis, just now one phase has been completed. Now, the energy payoff phase. They are producing 4 ATP. They are producing 4 ATP to NADH, and net gain will be 2 ATP. Now, see steps of glycolysis. We are having a 10 enzymatic reactions. Step 1, phosphorylation of glucose. The glucose at its 6th carbon place, one phosphate group has been added. So, with the help of enzyme hexokinase. But you should know, in the liver, this hexokinase is called as a glucokinase. Anyhow, both are going to add a phosphate group, and in this reaction ATP is consumed, and this reaction is irreversible. Once glucose will be converting into glucose 6-phosphate, it is irreversible. And why, for what reason this glucose will be converting into glucose 6-phosphate by the cell? See, whenever the glucose will be entering into the cell, whenever the glucose has to be enter into the cell, it should not enter into glucose acetase. It has to be in a glucose 6-phosphate. Only then it has been enter into the cell. Now, isomerization. Glucose 6-phosphate will be converting into fructose 6-phosphate with the help of the enzyme called the phosphoglucoisomerase, and both are structurally similar. And here, this is the second phosphorylation, that is fructose 6-phosphate will be converting into the fructose 1,6-bisphosphate. The most important enzyme will be the phosphofructokinase, PFK1. Very, very important enzyme. Very, very important enzyme. The ATP here, it will be consumed. So, this will be expected in our exam. Very, very important. Rate limiting and regulatory step, and this step is also irreversible. Once the fructose 6-phosphate will be converting into fructose 1,6-bisphosphate, it cannot back. It is an irreversible reaction. See, phosphofructokinase 1 will be the rate limiting and regulatory step. Why we are saying this particular step as a rate limiting step? Because of the enzyme phosphofructokinase. If there will be no phosphofructokinase, none of the other enzyme will be replacing this particular step. That's why we are saying this step as a rate limiting and regulatory step. Very, very important. Now cleavage. Fructose 1,6-bisphosphate will be converting into dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate with the help of an enzyme aldolase. I have explained to this. Now see this, the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate will be in an isomer. This is a reversible isomer. Both are structurally similar and the name of an enzyme will be triose phosphate isomerase because both are 3-carbon containing molecules. Now see, from this step onwards reaction occur twice per glucose. Per glucose reaction occurs will be twice. I'll explain, So the same thing. Oxidation and NADH formation. See here, glyceraldehyde 3-phosphate will be converting into 1,3-BPG with the help of this enzyme G3P dehydrogenase. So wherever you are seeing this particular enzyme, that is dehydrogenase, definitely there will be the production of this NADH. Wherever it is. Wherever you are seeing this dehydrogenase enzyme, there you can see definitely that particular reaction they are going to produce this NADH. Now this 1,3-BPG will be converting into 3-phosphoglycerate. Here, one phosphate molecule has been released. So here where the first ATP has been formed, and with the help of which enzyme? Phosphoglycerate kinase, and they are the one producing ATP. And here directly they are producing an ATP, and here the substrate level phosphorylation has been taking place. So we are having two types of ATP. Two types of phosphorylation. One is substrate level and another one is oxidative, and this will be the substrate level phosphorylation. And rearrangement here, 3-phosphoglycerate will be converting into 2-phosphoglycerate with the help of phosphoglycerate mutase, which means what? The third carbon containing a phosphate group in this. Here the phosphate group has been shifted to the second carbon, that's phosphoglycerate mutase. Now dehydration, which means what? Removal of a hydrogen. Removal of water, releases the water with the help of which enzyme? Enolase enzyme. 2-PG, 2-phosphoglycerate will be converted into phosphoenol pyruvate. Now this will be the final step, and in the final step the second ATP molecule has been produced. Phosphoenol pyruvate will be converting into a pyruvate with the help of which enzyme? Pyruvate kinase produces the ATP, and this reaction is also irreversible. And we have studied how many irreversible? 1. The step 1 is irreversible, and the step 3 is irreversible, and step 1. So 1, 3, 10 will be the irreversible step. So totally in the glycolysis we are having 10 steps. In the 10 steps, 3 are irreversible steps, all others are reversible steps. And what are all the 3 steps? First step, second, third step, and 10th. 1, 3, 10. These 3 steps are irreversible, and remaining all 7 steps are reversible. So this can also be expected. And which enzyme is called as a rate limiting step? Phosphofructokinase is a rate limiting enzyme. This is also very, very important. Now, what will be the overall reaction? See here, the glucose will be converting into 2-pyruvate, 6 carbon, and here 2 molecules of pyruvate, each pyruvate having a 3 carbon, 3 plus 3 is 6, equation balanced. 2 ADP, 2 PI, together they are forming 2 ATP, equation balanced again. Now 2 NAD plus will be converting into 2 NADH. So the end product will be 2H plus and 2 water. So that's all about the reaction of this glycolysis. So this will be the overview of the glycolysis. So at the 7th step and in the 10th step, 4 ATP, totally 4 ATP has been produced, and here 1 ATP and here 1 ATP has been used. So per glucose 2 ATP will be produced. As a result, they are also produced 2 NADH. So if we want to convert this NADH into ATP, 1 ATP it will be approximately equal to 3 ATP. So 2 NADH is equal to 3 into 2 to totally 6 ATP. So totally in glycolysis 8 ATP will be produced. Okay, 2 ATP, 2 NADH. If you want to convert this NADH into ATP, then 8 ATP. Don't confuse with this, right? Now, see the irreversible reactions are step 1, 3 and 10. Hexokinase, PFK, phosphofructokinase, pyruvate kinase. This hexokinase will be inhibited by the glucose 6-phosphate. And PFK1 will be the main regulatory step. Just now I have explained you. Pyruvate kinase activated by fructose 1,6-bisphosphate. So these three reactions are irreversible reactions. Now regulation of PFK1, phosphofructokinase. It will be activated by AMP, adenosine monophosphate, adenosine diphosphate, fructose 2,6-bisphosphate, but not ATP. So this phosphofructokinase will be inhibited by ATP, citrate and at the low pH value. Very, very important. What are all the fates of pyruvate? Now pyruvate has been produced as a result of glycolysis. So based on the oxygen availability, whether oxygen is present or not, if oxygen present, then this pyruvate will be converting into acetyl-CoA, and this acetyl-CoA will be enter into the T-cycle cycle, which will be in the mitochondria. And in the anaerobic respiration, this lactic acid fermentation case of animals, pyruvate will be converting into a lactate. Pyruvate will be converting into a lactate. And if it is yeast, there will be no oxygen. This pyruvate will be converting into ethanol and carbon dioxide, and this is what an anaerobic respiration. And these are all the important points. Just go through it. Now, that's all about the glycolysis. I think I have explained you all the most important keywords in the glycolysis. Now, come to the question, which of the following enzymes catalyzes the committed step of the glycolysis? Committed step of the glycolysis, that will be which enzyme? Rate limiting enzyme, that is none other than the phosphofructokinase. How many molecules of ATP are consumed during the preparatory phase of the glycolysis? That will be 2. 2 molecules. Which step in glycolysis produces NADH? So definitely that will be glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. So at this stage we are getting the NADH. Which of the following intermediates is the first to generate ATP during the glycolysis? Where the first ATP molecule has been produced? That will be in the first step, that is in the 1,3-bisphosphoglycerate. The net ATP gain from one molecule of glucose during glycolysis, net ATP gain will be the 4 ATP will be produced. Which enzyme is inhibited by the fluoride ions? That will be definitely enolase. Just make a note of it. Fluoride ions is going to inhibit the enolase. Substrate level phosphorylation has been taking place at which state? Wherever the ATP has been produced, that is nothing but a substrate level phosphorylation. That will be the 7th and the 10th step. And which molecule is the end product of the glycolysis under aerobic respiration? That will be none other than the pyruvate. And which molecule is an allosteric activator of phosphofructokinase 1? So I have explained you what is meant by allosteric enzyme whenever I explain you the enzyme and enzyme kinetics. Very, very important. Allosteric activator of phosphofructokinase. I have explained you allosteric activator here. AMP and ADP, these are all the activator. Only then it will be activate. ATP, citrate, they are the ATP and citrate, they were all the inhibitor. So who will be the activator here? Definitely that will be the AMP. Now, how many molecules of NADH are produced per glucose molecule in glycolysis? How many molecules of NADH? 2 NADH has been produced. So in today's class, I have explained you the basic information about cellular respiration. We have started that, and in that we are having 4 stages. 1 stage has been completed. Remaining step has been completed in the upcoming classes. Thank you so much. We'll meet you in the next class.